This is a fossilized mammoth tooth. It looks like a silent, static piece of stone that managed to survive for over 1,000,000 years. But if you grind a small piece of it into powder and extract the DNA, it is incredibly biologically loud. The sequencing machines will return millions of distinct genetic fragments. Some of those fragments belong to the mammoth itself. The rest is a chaotic mix of genetic material from plants, fungi, modern laboratory workers, and soil organisms colonizing it long after death. Buried deep inside all that material, there might also be traces of the bacteria that lived inside the mammoth while it was walking the earth. Normally, paleogeneticists treat that microbial DNA as an obstacle. When the goal is to sequence the genome of an extinct animal, researchers map the extracted fragments against a modern reference genome to isolate the mammoth DNA. Everything else is discarded as unwanted noise. A 2025 study from the Center for Paleogenetics in Stockholm entirely reversed that standard logic. The researchers removed the mammoth DNA, making that massive discarded cloud of genetic noise their absolute focus. But that data is a chronological mess, assembled over strata of geological time, containing host organisms, burial sediment, and recent contaminants. Because ancient bones are absolutely packed with bacterial DNA, the challenge is not finding it. The real scientific hurdle is proving which of those microscopic fragments actually shared a living body with a mammoth 1,000,000 years ago, and which ones arrived thousands of years later. To strip away the modern contamination, the researchers needed an evidence chain. The first filter in that chain is testing the physical condition of the DNA itself to prove its sheer antiquity. This diagram illustrates what happens to genetic material after cellular repair stops. Water, radiation, and temperature fluctuations break long molecules into shorter pieces. At the frayed ends of those fragments, one specific molecular base, cytosine, frequently loses part of its structure. Because of that chemical degradation, it is read by sequencing machines as thymine. That reliable cytosine to thymine substitution functions like wear marks on a vintage document. If you find a long, flawless bacterial sequence without any of this chemical damage, you are likely looking at a pristine modern lab contaminant. A highly fragmented sequence full of those specific chemical substitutions confirms the DNA is genuinely ancient. But physical damage has a fatal blind spot. A random soil bacterium that washed into the mammoth tooth 50,000 years ago would also carry that exact same ancient damage. The wear marks prove the DNA is incredibly old, but they cannot prove the organism lived inside the host. That requires a second filter, testing for spatial and temporal distribution. Because a single tooth can produce a misleading signal, the researchers leveraged the massive scale of their dataset, cross-referencing microbial DNA across 483 distinct mammoth specimens. An environmental microbe from dirt will appear randomly based on the surrounding soil. A lab contaminant will appear only in samples processed in the same batch, but a genuine mammoth-associated lineage will show a meaningful pattern. It appears repeatedly across different individuals, surfaces in specific tissues like teeth, and spans vast periods of time. Passing the tests of age and location narrows the field. To definitively lock these fragments to a living ecosystem, researchers must then place them into a rigorous evolutionary context. Analyzing ultra-short ancient fragments in isolation is dangerous. A few dozen broken DNA letters can easily look like sequences from many related modern bacteria. If you classify them one by one, the computer might assign the fragment to the wrong species entirely, simply because that's what happens to be available in a modern database. As we see in this animation, the researchers solved this by finding genetic differences shared across many sites and linking them together. By finding overlapping sequences, they rebuilt partial bacterial genomes out of molecular debris, much like taking thousands of damaged copies of the same book and aligning overlapping fragments of text to rebuild whole pages. Once they assembled these larger structural blocks, they mapped them against vast evolutionary family trees of modern bacteria. Instead of a loose guess based on a single fragment, they could verify whether these reconstructed sequences formed their own coherent ancient branch. After millions of fragments were filtered for chemical damage, checked for broad distribution patterns across 483 mammoths, and subjected to strict phylogenetic placement, Exactly 6 microbial lineages survived every test. The oldest reconstructed signal came from an Erysipelothrix-related sequence found inside a steppe mammoth that lived approximately 1.1 million years ago. That represents the oldest authenticated host-associated microbial DNA ever reported. Other surviving lineages offered equally tantalizing details. The team found a Pasteurella bacteria closely related to a strain that causes fatal blood infections in modern African elephants. They also found a Streptococcus lineage anchored specifically in the teeth, persisting within mammoth populations across massive intervals of time. But biological relatedness is not identity. Bacterial genomes gain, lose, and swap genes extensively over a million years. Because these are still incomplete ancient genomes, without soft tissue evidence or clinical observations, it is impossible to state definitively whether a mammoth carrying these microbes was completely healthy or suffering from a fatal infection. The picture we get from this data is strictly limited by preservation distortion. The fossilized materials themselves dictate which microbes survive long enough to be sequenced. Dense bones and teeth are excellent at protecting DNA from the elements. Because of that, microbes recovered in this study are heavily biased toward oral and blood-associated organisms. Traces of the bacteria confined to the mammoth's gut or skin decayed beyond recognition long ago. To be incredibly clear about the boundaries of this science, no living bacteria were revived. The volume of fragments found does not equal the original population size of the microbes. And there is zero evidence suggesting these specific bacteria contributed to the extinction of the mammoths. By combining chemical damage patterns, broad sample distribution, genome reconstruction, and phylogenetic placement, the team engineered a chain of evidence separating biological reality from noise. This allows scientists to observe how specific microbes adapted alongside their hosts across hundreds of thousands of years. A fossil preserves a host and its colonists simultaneously. It is the damaged record of a million-year-old ecosystem existing inside a single animal.